to do all of this through A, B, and C, and D. Huge problems. I won't go into all of the problems because I could spend probably 5 hours just listing all of the issues. Repetitive sequences in the genome. I'll just note a few here. Which gene model? Do you think we would know already where the genes are in a human genome? We don't. We're still discovering new genes. So this is called gene modeling. We don't even know when we get our data what to compare it against because it's a moving target. And when I say moving target, the genome assemblies change about once a week to once a month. So this has to be something you do on a regular basis because they're updated. They're competing ones. There are ambiguous reads. We're seeing introns that should have been spliced out now in our sequences. What do we do with all this? So data analysis nightmare. What I'm going to show next comes from this, a little bit old now. 2011 is just incredibly old in this field, but it really delineates in a very clear way some of the problems with RNA-seq analysis. So I have, and I think you post the PowerPoints online, so I'll have that up there. Okay, first of all, alignment of reads to the genome. And this is where now mathematicians need to get involved. Every couple months there's a new way of doing this, a new computational method. There's no accepted method in the field yet. So this is really an exciting and emerging field. There are a couple different ways, and this is the first thing we want, to kind of see what we have after this analysis. An exon first approach, where everything that maps to a known exon, and we don't know where all the exons are either for sure, is first mapped and then they're extended against the gaps there. Another method that we'll talk about more in a minute is to take all of the known sequence, divide it into what we call k-mers. So choose your k. It could be k of 5, it could be a k of 10, a k of whatever, and then use those as seeds to extend across. There are problems with both of these approaches. And in the paper, there are references to some of the basic mathematics that is used for those. Once you have an idea, then you want to get the transcriptome. This is the idea. So RNA-seq is also called transcriptomics because transcription goes DNA to RNA. The transcript is the RNA, so we call RNA-seq transcriptomics as well. So you want to get what we call transcriptome, all of the RNA expressed in that cell. Many different ways of doing this as well. They fall kind of into two categories here. A genome-guided approach. This is now more in disrepute because there are a lot of problems with what comes out of that particular way of doing it. And these de Broglie graphs and using graph theory to come up with, again, using these k-mers, that you take the genome or the transcriptome and divide it into all the possible pieces, make a usable data structure out of that, and then any of the overlaps will actually be the nodes in the graph, and then you use basically an assembler that tries to identify this Eulerian path through the graph. That's the current method of doing this. Once again, a lot of interest in improving this because right now, nothing is working that well. And the same group will do the same experiment, analyze it through different computational methods, and really get totally different results. This is a problem for the biologists. We also want to know the quantification. How much is there? How much of a given gene? This seems like it would be fairly simple, But once again, huge computational problems here. How do you do this? If this is looking, for example, at 2 different RNAs here, and these are the reads, how do you figure out, how do you count, and which do you count if you get a few reads over here for this particular exon, a lot of reads over here? These reads don't go together, so how do you know that that read actually goes with that one, or that one, or that one? You just don't know. All kinds of statistical maximum likelihood approaches being used here. None really perfect, and each one giving very different biological answers to the question of how much is there. You think it would be very straightforward, but not when you're dealing with this splicing issue and so many millions. And we're only talking about one experiment now. This is just looking at one. So some approaches look at what is common to all the exons. Others look at, so intersection, there's union, many different approaches for getting the quantitation. Again, nothing is perfect. A field that's wide open. We have no idea how to normalize these appropriately because every run is different, every sample is different. We put in reference genes when we do this RNA-seq, but those aren't always the same from run to run. So even normalization, something that we've been dealing with as biologists for hundreds of years, even that's not worked out because of the massive data for RNA-seq. And here's what we really want to know at the end of this. What are the genes that are different? What's in that cancer cell that's not in a normal cell? Or what's in the normal cell that's not in a cancer cell? What's in a regenerating cell? Why could that tadpole regenerate brain cells and we can't? What genes are being activated? So unlike old technology, microarray technology, which was just based on relative fluorescence, had a very small dynamic range as well. Another problem with this data, for RNA-seq, the range goes from 1 to hundreds of thousands of transcripts. So orders of magnitude that the various computational techniques have to be able to deal with. So yet another problem. Here we have these discrete reads, and this is trying to come up with an approach that will be able to say, here's what is different. After looking at the reconstruction of the transcriptome, of the transcripts, here's what's different in each of these different techniques and cell types. This area is wide, wide open. I mean, this one, there's no agreement at all because it's even at a higher level of analysis than, okay, how many, or okay, well, what is there? You know, is this an isoform or is that an isoform? So the differential expression where you're comparing two very different experiments is really, really difficult. And this isn't even dealing with the N problem. So in any science class, psychology class, social science class, first thing you learn is you have to repeat your experiment. The more times you repeat it, generally the better confidence you have in the analyzed results. Well, these are expensive experiments, and they're really hard to do as well. So the N for these kind of RNA-seq experiments is generally 2. That's what's accepted in the literature. How do you do statistics on an N of 2? Big problem. So very few of these have more than 3 or 4, unheard of. And then how do you compare and normalize among experiments? You have no idea, because these data are just emerging. Okay, that's all the easy part. How do you interpret the data? How do you make sense of it? Biological. You know, what we want to know what is different, the pathways that are different. There's not going to be one gene that's different in a cancer cell compared to a normal cell. There are going to be thousands of genes, tens of thousands of genes, but very small, subtle little differences, because one gene will regulate, and a protein will regulate the expression of another gene and another pathway. So this really is the biggest challenge. Once we could agree on all the data analysis, just getting kind of the facts of the RNA-seq experiment, how do we make sense of this? So we want to group these genes into something biologically meaningful. We also want to visualize it. We can't look at 100 million different sequences and make sense of it. So one thing that is being done in the field, and this is a real mess, we started dealing with this in the lab, something called gene ontology, where every single gene that has been sequenced on the planet for any organism is associated with a few tags. Where it is in the cell, what its biological process is, and what its molecular function is. But these are overlapping, sometimes totally meaningless. The hierarchies don't make sense. There's not a clear unidirectional hierarchy. They could go in all kinds of different directions. You try and use these Go terms. Sometimes a gene will have all three tags for the process, the function, the component. Other times it will have just one or two. So you really can't use that for your analysis easily. This field is in huge need of some computational statistical analysis because it's really, really, really messy right now. And we desperately need this because we can't go through all these genes by hand and associate function. We need to do this using some kind of good algorithm. Anyway, when we tried doing this, it was just impossible to get any kind of biological information meaningful out of it, because if you can't be assured that your gene will have at least some of each of these categories, if it's missing, that will look like that category is not being expressed in the RNA-seq. A lot of people are working on this from a mathematical computational point of view. We want to take all that, the information that is associated with each gene, and this is what we really want to do. Pathway analysis. Do we see that a tyrosine kinase pathway, this is an enzyme that makes cells divide, is associated with a cancer cell? And what we would do then is not try and knock out just one gene or protein in there, but really target many points of that pathway. So this is going to be very useful in therapeutics. It's also going to be useful to say, okay, these genes, these pathways are what are allowing a particular tadpole or fish to regenerate and us not. So you can see here, these are some of the Go terms, but they're kind of mixed up. And these arrows are indicating, they could indicate a lot of things. First of all, that those two are always expressed together, but we want more than that. We also want, well, this particular protein regulates that one. So we not only want a spatial pathway, a snapshot in the cell, we want at least a four-dimensional one where we see which of these proteins is regulating other downstream biological events in a given cell type. So this obviously is just in its infancy, developing finally now as we have these global expression techniques. So I'll just end again with some of the problems that we're facing here in the RNA-seq field. And what I love that quote from the call for our NIH, grants spectacular opportunities for you mathematicians out there. Biologists really, really need you. I'll end with that. Thank you. And if you ever have any questions, I live over in ISC on the 3rd floor. Come talk to me anytime, and I'll be happy to discuss our projects and data with you. We have ongoing collaborations with the math department. And come to the biomath lunches. The food is great. The science is even better.
So before you go, just a reminder that this Friday we have a talk, so and after talk we'll have a reception in the math department also. So please be here. So the talk in the small 110, okay? And the speaker will be Don Brown from UVA, Computer Science Department, and he will give, I think, very nice overview, abstract looks like.